we will continue the same topic, proteins. Now we will study some more properties of proteins. Proteins are extremely reactive. They readily react with any substrate, quickly react with the substrate, so they are extremely reactive and highly specific in behavior. They are specific in behavior, that is, a specific protein is required for a specific reaction or a specific place, purpose, whether it might be structure or hormone or any other function. So a specific protein is required for a specific function. Proteins are amphoteric in nature. Amphoteric means they act both as acids as well as bases, because they contain the acidic group, that is the carboxyl group COOH, and the basic amino group NH2 group. As we already studied the structure of amino acids, in which there is acidic group as well as basic group. So proteins, they are amphoteric in nature. They behave as well as acids as well as bases. The behavior of proteins is strongly influenced by pH. If the pH is more or less, so the behavior of protein, it is changed. It becomes acidic or basic, and it is neutral with a neutral charge at an isoelectric point. So like amino acids, proteins are dipolar ions. Dipolar ions means when proteins, they are placed in the aqueous medium, so they get converted into the Zwitter ions. Zwitter ions are the ions of amino acids which contain a positive charge and a negative charge. The charge is because of the movement of the hydrogen atom from the carboxyl group to the amino group. As the amino acids are placed in the aqueous medium, there is transfer of one hydrogen atom from the carboxyl group to the amino group. So the carboxyl group it gets a negative charge, and the amino group it gets positive charge. So this negative and positive charged amino acid ions, they are called as dipolar ions, or they are called as Zwitter ions. So these Zwitter ions are produced as at the isoelectric point. Isoelectric point is the pH, where it is the pH at which the amino acids, they carry no net charge, because the number of positive charges is equal to the number of negative charges, and the net charge is 0. The ionic groups of a protein are contributed by side chains of the polyfunctional amino acids. So the amino acids, they contain ionic groups, and these ionic groups, they are contributed because of the side chains. A protein, it consists of more basic amino acids such as lysine, arginine. Lysine and arginine, both these are basic amino acids. So when a protein, it contains more basic amino acids, so they behave as positively charged ions at a physiological pH of 7.4. Means at a pH more than 7, they will behave as basic amino acids. Such proteins are called as basic proteins. The proteins which contain basic amino acids like lysine and arginine, then the proteins are called as basic proteins. For example, histones of nucleic proteins are the basic proteins. Then a protein which is rich in acidic amino acid exists as an anion, that is negatively charged ion, at a physiological pH less than 7. Everyone of you all know that when the pH is less than 7, so the substances, they are acidic. So at the acidic pH, the amino acids, they will be negatively charged anions. Such proteins are called as acidic proteins. Most of the blood proteins like albumins, globulins are acidic proteins because they contain the acidic amino acids. These charged ions, cations and anions, they are produced in the aqueous medium. Proteins, when they are placed in aqueous medium, they form ions like positively charged ions or negatively charged ions because of the type of amino acids they contain. If they contain basic amino acids like lysine, arginine, so they will get converted into positively charged ions. And if they contain acidic amino acids, then they will get converted into negatively charged ions. So example of basic proteins are histone proteins present in DNA, RNA, nucleoproteins. These are basic in nature because they contain basic amino acids. And the proteins which are present in the blood like albumins and globulins, they are acidic in nature. Now the classification of proteins. On the basis of their structure, proteins are classified into three categories. First type is the simple proteins. Simple proteins on hydrolysis, they yield only amino acids. When the proteins are broken down, you will get only amino acids, then those type of proteins are called as simple proteins. Simple proteins, they may be soluble in water. For example, histone proteins, they are soluble in water. The histone proteins present in the nucleic acids, they are soluble in water. The globular molecules of histones, they are not coagulated by heat. As you all know that when the proteins are heated, they are coagulated, means their structure is changed and their function is also hampered. But the globular proteins of histones, they are not coagulated by heat. It is their characteristic feature of histone proteins. Then albumins are soluble in water, but they get coagulated on heating. So albumins which are present in the blood, they are soluble in water, but they get coagulated on heating. Coagulated means their structure is broken down and they form a clot-like structure. Albumins are widely distributed. For example, in egg, there is egg albumin. In serum, blood, there is serum albumin. And in the pulses, there are albumins which are called as legumulin. Legumulin word has come from legumes. So the albumins which are present in legumes are called as legumulin. Next type of proteins are conjugated proteins. Conjugated proteins consist of a simple protein united with some non-protein substance. So the amino acids, they are united with some non-protein substance. This non-protein substance, it is called as prosthetic group. For example, hemoglobin. The hemoglobin protein which is present in the blood, it is an example of conjugated protein. Globin protein, it contains the iron containing pigment hem, and because of this hem, the blood is having the red color. So the hem pigment, it contains iron and the protein is called as globin. So hem is the prosthetic group as it contains iron, and this is the hemoglobin, example of conjugated protein. Similarly, nucleoproteins have nucleic acids coupled with histone proteins. So in nucleoproteins, the proteins are histones and the nucleic acids, they function as the prosthetic group. Then glycoproteins. In glycoproteins, these are also conjugated proteins in which the sugar plus protein molecules are present. Mucoproteins is also a type of glycoproteins. Mucoproteins, they are the combination of carbohydrate and protein complexes. Example, mucin present in the saliva. The mucin protein which is present in the saliva, it is a combination of carbohydrate and protein. As you can taste that the saliva in your mouth, it is a bit sweet in taste because it contains sugar and protein. Then heparin of blood. The heparin protein which is present in the blood is also an example of mucoprotein. Lipoproteins. Lipoproteins are lipids plus protein. So lipoproteins are the lipids plus protein complexes. These are present in the plasma membrane in high amount. Lipoproteins are present in plasma membrane in the cell. Then they are also found in the brain. Some conjugated proteins are found in the brain. In milk also you will find the conjugated proteins. The last type of proteins are derived proteins. In derived proteins, these proteins are not found in nature. These are derived from the native protein molecules on hydrolysis. So the molecules which are present in some naturally occurring substances, those are broken down, those molecules are broken down and the derived proteins are formed. The first example of derived proteins are the metaproteins. Now these metaproteins are organic compounds which result from the reaction between an acid or alkali and a protein. A protein, it is subjected by an acid or an alkali. Now, and you get some compounds, and those compounds are the metaproteins. Now they are soluble in weak acids or alkali and they are insoluble in neutral solutions. For example, albuminate. Albuminate, it is obtained from the hydrolysis of albumins. Then peptones are derived proteins. Now peptones, these are protein hydrolysates. Hydrolysates are produced by hydrolysis of some compounds. So proteins are hydrolyzed, that is broken down with the help of water molecules, and they are formed by enzymatic or acidic digestion of various raw materials, and their media act as the stores of nitrogen. For example, the casein hydrolysate from milk. From milk, this peptone it is formed, that is casein hydrolysate.